bad. Sorry about that. I think we're good. Can anybody hear me on the Zoom? Okay, thank you so much. Yeah. Welcome everyone. Thank you for coming. Today we are going to be hosting two wonderful speakers, so let's get started. For the online folks, please mute your microphones before we start. And this event is being recorded, so if you have any questions, please feel free to reach out to Will by this address. Today we're going to be starting with the announcements that I'm making right now, and then the introductory trainee talk will be taking place. After that, we'll be having our main feature talk, and after the finish of it at 7:20, we will be having the social networking session. WEMBAC organizing committee is comprised of 3 faculty members, Dr. Williams Howe, Dr. Amy Lee, and Dr. Faraz Haj, and wonderful volunteers as well. We have 2 more WEMBACs coming up on March 19th and April 30th. Please check out our website for more details. If you want to subscribe to our mailing list, you can go to wembac.org, click on this contact here, and then fill out the form under the mailing list. If you don't want to miss out on the events that we are holding here, you can also use the following links and social media accounts to follow us. And if you want to suggest a speaker for the next season, starting next year, please go to the contact tab and use the suggest a speaker button to do so. We are sponsored by Graduate Studies in Bioinformatics at the University of British Columbia and Simon Fraser University, Genome British Columbia, SFU Big Data Hub, Michael Smith Laboratories at UBC, Langara College, Stem Cell Technologies, Vancouver Prostate Center, and we are also affiliated with International Society for Computational Biology, Montreal BUG and Toronto BUG, and we are a proud partner of the Canadian Bioinformatics Hub at British Columbia node. So for the Zoom audience, please ask your questions in the chat, and one of the volunteers will be voicing it for you. Today we are going to start with Maxwell Douglas. He's a PhD student in bioinformatics at Park Lab at the University of British Columbia, and the talk title is Hunting for Mediation in Expression Quantitative Trait Loci, a case study using ovarian cancer.
Cool. All right. Thank you very much for the introduction, Berke. This will be a presentation on a piece of part 1 of my thesis. So the title for my talk is Hunting for Mediation, a case study using ovarian cancer. Ovarian cancer is the primary disease of study that I am looking at. The agenda, I'll be going over 4 concepts as kind of background information for the talk first, and then hypothesis, methods, and some results. So I'll begin with germline variation. In the department that I'm in, a lot of people look at somatic variation predominantly, and that's for a good reason. In epithelial ovarian cancer, the most deadly form, and there are subtypes even within that, high-grade serous ovarian cancer is the most deadly within the epithelial subcategory. Somatic does make up the majority of the risk. It dominates the risk, but there's about 40% that is familial. It's germline risk. This risk can be also broken into pieces for what we've explained and what we understand and what is yet to be figured out. So about 27% of that can be attributed to the common BRCA1, BRCA2 variants, another 13%, other fairly high, moderately penetrant genes, a lot in the homologous recombination pathway, and then there's about 6% that have been identified thanks to GWAS studies. These are common variants. And then there's a massive proportion, over half, that is unexplained. and this forms what's called the missing heritability category. This is a common category across a lot of different traits. For traits that are extremely easy to study, like height and weight, etc., we can have millions of data points there because it's easy to evaluate people that way. As long as you have a genome panel and access to the patient, then you can get that kind of information. And so the missing heritability there is a lot smaller. But for a lot of diseases, it's a large proportion. So a bit more on genome-wide association studies. Why we do them is because they allow us to discover common germline variants associated with a trait, and you do this by leveraging large sample sizes. It's a case control style of study. So you have a bunch of genetic variants, a SNP at one location or another, and you compare the frequency of that SNP in cases versus controls. You can then model this. A simple one would be using a linear model. So the trait here would be T, X would be the genotype, and you're finding your betas plus an epsilon for error. You can then get p-values from this, and this is a common way of looking at this graphically, because you get a lot of data out of these kind of studies. So to plot the negative log 10 p-value along a genomic axis, so along the x-axis there you have all your chromosomes, 1, 2, 3, all the way through the x and y. And what's raising off of the x-axis are the most significant p-values, or the most significant loci. These are loci that are most associated with a trait. So for us, the trait is ovarian cancer, and we're trying to find SNPs associated with ovarian cancer. The next bit is quantitative trait loci. These link genetic variants to molecular traits. So not quite the same thing as GWAS. We're looking at molecular traits this time, and it's highlighting a potential mechanism for how that locus affects the biology. So for expression quantitative trait loci, you're looking at expression. That's the quantitative piece. Gene A in this panel is higher without the locus than with the locus. The locus is that lollipop thing that you see sticking off of the x-axis there, and it's affecting gene A. There are other types of QTLs, though. Splicing QTLs, isoform QTLs, protein QTLs, polyadenylation QTLs. Many different options here for trying to trace the path from a SNP to its effect. You can combine these two types of data points into something called a TWAS, or do colocalization analysis. They're kind of more powerful together. And this suggests a path by which, you can use this to suggest a path by which a GWAS hit could affect a trait. So if you have, it's exactly, in the case of colocalization, it's exactly what it sounds like. If two SNPs are in the same place, or even the same SNP, and one affects the gene expression in one data set, and in the other data sets it's associated with the disease, you're making an association that perhaps that gene expression is an intermediary between the trait and the genotype.
Sorry, is this informatics meeting?
Yep.
Oh, it is?
Yep.
Oh, okay, I have a couple of people. I'll send you.
The next option is TWAS. In TWAS, it's a little bit more complicated. First, you train an expression model using a paired data set of genotype and that molecular trait that you might have. So this is usually in the same type of data set that you would find your QTLs. So in the expression there on the right-hand side of the slide, Y is the expression, and you're learning the W's, and X is, again, the genotype. You then take these weights that you've learned, and you try and impute the expression in the GWAS cohort. So X, GWAS, this is now genotypes that haven't been, or you haven't seen yet, but are hopefully, there's a fair amount of co-occurrence is the right word, between the GWAS and the genotypes that you got from the training set. And then you test the imputed expression for association with the trait. So, long story short, quantitative trait loci, or QTLs, are worth identifying. But as you might already suspect, they're of limited use without further annotation. So this might be done in one tissue or another tissue. The type and how you've done the QTL study matters. Many, many pieces of this matter. So what additional information can be added to these QTLs to make them more informative? I'm interested in mechanism here. Transcription factors could possibly mediate the effect of a SNP on a molecular trait. So X, again, the SNP or the genotype, Y, the expression, we might have a mediator in between the two. So C is the QTL here, that edge is the QTL, and then you're looking to see if the edges A or B exist. So the hypothesis. That paired RNA-seq and genotype data can first be used to map QTLs, and then second, identify instances where a third element, a transcription factor, mediates the effect of a SNP on expression. So we built a pipeline to identify possible mediation of QTLs across relevant tissues for high-grade serous ovarian cancer. For example, for ovary, in the GTEx cohort, we had 167 samples. We included covariates for genotype, peer-inferred expression factors. And then we mapped QTLs using an existing method that we heavily refactored to make it massively parallel and allow in-flight random sampling of QTLs. This is kind of design choice there. And then we performed a wald test between the base run of the tissue and then when adding additional covariates. So we added a transcription factor. And in this case, we used pan-cancer transcription factors, so just common transcription factors associated with cancer. A wald test is the simple approach. So at first, we did the simple approach. We were able to identify hundreds of thousands of QTLs across OC-relevant tissues. This wasn't super surprising. This has been done many times, but as a first step, great. Our p-values were mostly as expected, nice and uniform, and then they were the observed versus expected in QQ plots was fairly aligned until about 1 when it departed from normal, and that's also as expected because we found many, many QTLs. So the QQ plots look as expected. However, we found no mediated genes, no instances of mediation across any of the tissues. However, will a more powerful method, so something like Bayesian mediation via the causal steps approach, be more successful? In this design, for each triplet, so a QTL, transcription factor, a target gene, we evaluate 8 competing causal models to find by which edges are present versus absent, and each model specifies a different directed acyclic graph over the 3 variables. And then we compute likelihoods using marginal multivariate t-distribution and conditioning on covariates. On the right-hand side of the slide, you can see the different models. I've labeled 4 and 8. The 4 case is the partial mediation example. The 8 case is the full mediation example. So in 8, you can see the edge between X and Y isn't there. And we're evaluating the probability of mediation given the data, and probability of that 4 model given the data plus the 8 model given the data. To do this, we used the B-mediator software. This has been implemented in that piece of software. So we wrapped it, added it into the pipeline, and then also theorized about some additional transcription factors. Because as I mentioned earlier, in the first iteration, we just tried a general set of pan-cancer transcription factors. This time we tried, or we also added some more tissue-specific transcription factors. So ones for ovarian identity, hormone receptors, and the EMT program as well. And that's where I have to end because this is ongoing work. So that's what's been done, and things are actually running right now. So it's on the compute cluster. Thank you very much.
I think we have 5 minutes for questions. If you have any questions, feel free to ask, please. Can you please touch on the red button? Yeah.
What do you mean when you say you add a transcription factor? Could you explain what you mean by that?
Sure. Is my thing still up? Perfect. Let's go back to here. Okay. So on the far right, you see the bin with covariates, principal components, pure factors. We added the expression values for a transcription factor across all the different samples to that covariate matrix and just accounted for it.
Please join me in thanking Max for his presentation. Now we are going to have our featured talk by Dr. Anne Condon. I would like to talk about her background a little bit. Anne Condon is a professor in computer science at UBC, where she has served as department head and associate dean in the Faculty of Science, trained as a computer science theoretician. Her current research interests are in biomolecular computation and computational prediction of RNA and DNA reaction kinetics. Anne received her bachelor's degree in computer science and mathematics from University College Cork, Ireland in 1982, and her Ph.D. in computer science at the University of Washington in 1987. She's an ACM Fellow, a Fellow of the Royal Society of Canada, and a recipient of the Computing Research Association's Habermann Award for her work on increasing the numbers and successes of women in computing research. And her talk is named Some Hows and Whys of Computing with DNA Molecules. Please join me in inviting Dr. Condon to the stage.
Thank you, Berkey, and thanks, Maxwell, for a very fun talk. I hope you'll give us an update in 6 months or a year. And thank you, everyone, for coming. It's really fun to be back giving a Vanbug talk after many years. And I figure many of you are not that familiar, maybe, with the field of biomolecular computing, so I thought I'd start with a little bit of an overview, and then I'll go in a little bit more depth to some specific research that our lab is doing right now on predicting DNA reaction kinetics. Okay, so what is biomolecular programming or biomolecular computation? So I usually like to describe it as the opposite of bioinformatics, which makes it kind of fun that I'd be invited to speak at a bioinformatics seminar. And it's the opposite because bioinformaticians are using standard silicon technology to understand molecules, and biomolecular programmers are using DNA as a raw material to solve computational problems. So just say as a sculptor might make something beautiful out of a piece of wood, but wouldn't have a clue about like forestry, that's the way we are. We don't really understand biology necessarily or DNA, but we think of it as an amazing material that we can do new and creative things that nature might never have thought about. So it's also not really synthetic biology because synthetic biologists tend to harness the molecular machinery of the cell to do very creative and wonderful things. Whereas people in my field do more bottom-up engineering. They start with the raw material and they're building interesting things from scratch. And I would like to add that it's a super creative field with many programming modes, including chemical reaction networks that I'll mention a little bit, DNA origami, which you might have heard about, and I'll show a picture later, DNA assembly, and more. So there's many different, completely different modes of computing with DNA molecules. I got interested in this field back in 1994 when Len Adelman, who is a computer scientist, probably best known as the A in the RSA crypto system, he went into a wet lab and solved a very simple combinatorial problem, sort of like the traveling salesman problem. And I was at that time kind of ready for something new. I had just gotten tenure at the University of Wisconsin, and I decided if I was going to work in this field, I wanted to collaborate. So I found some amazing chemists that would work with me, and we worked on this for a long time, culminating in a paper in Nature just after I got to UBC, actually. But I would say, well, this was really fun, and it was a great way for me to get my feet wet in this area. Solving combinatorial problems in a test tube isn't really going to get us to an interesting place, because we can solve combinatorial problems just fine on silicon technology. And it's where computing in a wet environment or in a cell really is where there could be a lot of potential for computing with molecules. So a paper that really inspired me that appeared in 2010 by David Soloveichik, Georg Selig, and Eric Winfrey was a paper that said, hey, we need a kind of a high-level programming language to think about what we can do with molecules. And they suggested that we harness chemical reaction networks, which have traditionally been used by chemists to describe extant chemical systems. But what if you could invent your own molecules and invent how they interact with each other? What kinds of things could you get the molecules to do? So in this very simple example illustrated here, the molecules are simply functioning as oscillators. And the chemical reaction network on the left is sort of your high-level programming language. And in this paper, they showed that you could compile it down to a lower level DNA implementation, and then you can use various technologies like fluorescence detection to read out the results. There is a beautiful theory of what you can compute with chemical reaction networks. Everything from like simulating Turing machines, again, which you wouldn't want to do, but it's just proof that chemical reaction networks are capable of universal computation. One recent highlight that I really like is chemical reaction networks that can simulate rational weight rectified linear units. So they can kind of do simple neural network type pattern recognition, and that kind of computation could be put into a cell. And this is particularly nice because these chemical reactions that they're using are rate independent. So you don't have to, when you compile it down, be very worried about getting the rates exactly correct. I would say that the compilation process, once you've come up with a nice theory and you know what you want to implement, compiling it down to DNA is obviously very hard, and it's an area that's in work in progress. But what I'm going to do next is tell you what DNA strand displacement systems are and how, one way that you could take a chemical reaction network and synthesize DNA strands that would represent the abstract molecules in the network and then perform the computation that's encoded in the chemical reaction network. So DNA strand displacement is quite simple. You've got 3 strands, and the strand on the top, which we often call the invader, it has a toehold, a short sequence of DNA nucleotides that are complementary to a toehold on the substrate. So it can come in and bind to the substrate, and then a process called branch migration can cause it to bind to the substrate and force a long domain of the other strand called the incumbent or the target off. And at that point, that strand is only bound by a very short toehold and it can fall off. So one strand comes in and displaces the other. And in this particular case, because there's toeholds at both ends, the system is reversible, but you could remove the toehold on the right end if you wanted to discourage the reverse reaction. Okay, so now how do we compile chemical reactions down to DNA strand displacement systems. So a very simple reaction is molecule A plus molecule B should produce molecule C. And so I've got 3 strands here, colored strands that look in kind of a weird shape, but ignore the shape. It's just convenient for where I'm going. There's nothing special about it. And in order for this reaction to happen, what we're going to do is we're going to have extra molecules that are kind of like fuel molecules, and those ones will be in relative high concentration compared to the signal species A and B. And this big fuel molecule has one long bottom strand, and then it's got 3 shorter top complementary strands that are bound to the bottom strand. And then there's one extra little player there off to the right. So first of all, the A molecule can come in and bind to the free toehold. A process of branch migration takes place, and then a new toehold is freed up. At that point, the B molecule can come in and bind and force another toehold off, and all these toeholds are different, so things are very prescribed. And then finally, the extra small fuel molecule can come in, and it can force another branch migration, forcing off the molecule on the right, which is our product C. So you could, of course, elongate the fuel strand at the bottom so that A plus B could produce 2 product molecules, for example. So this is how you could take your high-level reaction or one instruction in your CRN program, and you could implement it in DNA. It's not the only way you could do it, but it's one way that you could do it using DNA strand displacement. And yet other authors have tried to figure out how would you, if you wanted specific rates, how would you control the rates of the different reactions that you would like to have happen in your test tube. Okay, so I am not going to talk a lot about applications today, but usually people are interested, so I thought I would put in one slide about applications. I would say one of the great things about this field is we don't really know yet what the applications will be, which means that we're all focused on the science and we're not focused on the money. So I really like that aspect, and we have to collaborate with each other. It's really fun. So one example is a paper from Peng Ying, who's at Harvard, and he is kind of a biomolecular programmer, but his work actually is very broad. But he was interested in cellular logic computation using RNA, so not DNA, but RNA. And so in this paper, he says he's reporting on a strategy for constructing RNA-only nanodevices, sort of like RNA switches, which many of you might have heard of. And these switches can be combined to evaluate complex logic in living cells. And in the picture in the bottom that I took out of the paper, it's a simple OR gate. So if either signal A or signal B is present, then the ribo switches change conformation and a gene can be expressed, for example. But you could imagine much more complicated logic that could control the expression of the gene. And so what they're doing is saying we can design computing devices that then could be used in a cell. And this is a small example of the potential here for this type of work. Okay, so there are these amazing scientists all over the world going into labs and doing biomolecular computation. I'm in a computer science department. I don't have a lab, but there are lots of supporting computational methods that are needed to do this kind of work. So as I said, there's a lovely theory of what you can compute with chemical reaction networks. That's very theoretical work. Then you have to figure out what are maybe better ways to compile down from chemical reaction networks to DNA strand displacement systems than the simple one I described. Then you've got all these DNA molecules, and they're going to have structure. They can fold on themselves. They can do all the kinds of things that you never thought they were going to do. So you'd better be able to see, are they actually going to fold into the structures that you thought they would once you designed them. And so DNA secondary structure prediction is valuable and important. And beyond that, since these chemical reaction networks or DNA strand displacement systems are very dynamic, you want to know about the kinetics, the folding pathways, as well as the equilibrium structures. And finally, if you can do prediction work computationally, ultimately you would like to build that into design tools that could design molecular systems to do interesting things. So I thought I'd mention that back in 2003, I gave a talk about work that our lab was doing on DNA secondary structure prediction, and I was able to pull out a photo that reminded me of the good old days. Sonia, who's here, was in the lab at the time. She wasn't actually working with me, she was working with somebody else, but we really did have a good time back then in the lab. And Hosna wasn't in that picture. I think Hosna gave a talk a year or two ago here at Danbug. So I put a little picture, a little inset picture of Hosna. And there were a lot of babies in the lab at that time too, if I remember correctly. So I had to make sure we got a picture with a little baby in there. So that was all work we did quite a long time ago in our lab. And now what I'm going to talk about today is more work on DNA kinetics prediction. And again, there's a whole bunch of a new generation of students who are doing this work. Nassim on the left, Mark Schmidt is actually an ML professor in our department. Boyan, Chen Wei, and Jordan are shown in the picture in the middle. Chen Wei actually just graduated, but Boyan and Jordan are really doing the hard work that I'm presenting today. And we collaborate with people at Caltech, including Eric Winfrey, who are they're wet lab people who can then hopefully use the tools that we build. So the rest of my talk is on the problem of computational DNA reaction kinetics and how we address this problem. And I hope that from the first part of the talk, you can maybe see why this would be interesting and useful to people in the field of biomolecular computation. And I'm going to do 4 things. I'm going to give you a couple of motivating examples so you kind of understand what the problem is. Then I'll describe what typical methods are. Then I'll talk about how we're trying to improve those methods, and then where ultimately we'd like to take this if I had another 50 years, which I probably don't, but we'll see. Okay, so DNA reaction kinetics prediction, you've got input and you've got an output. The input is going to be a description of a reaction. So three-way strand displacement is a good example. You start with the invader and you've got a target bound to a substrate. And after the reaction completes, you've got your products. The target has been released and the invader is bound to the substrate. So that's your reaction, and the input would be the sequences that are involved. What are the reactants and what are the products? And usually we would describe those in terms of secondary structure, which base pairs are bound initially, which base pair should be bound at the end. So that's a reaction. And of course, it might not happen at all the way you think it would happen. Like maybe the target will fold into a hairpin structure or some weird things will happen. So, you know, the purpose of DNA kinetics prediction is to see like what is the folding pathway that will get you from start to end, and will you actually get to where you want to go if you put those reactants into a test tube. So the most basic thing you might want to know is, well, what's the rate at which the reaction takes place? Or, you know, how much time would you have to wait for, say, 50% of your molecules to have completed the reaction? So that would be the most basic kind of output you'd want. So in my first example, I'm looking at data from a paper that studied DNA strand displacement. And these people were interested in, well, we know what DNA strand displacement is, but what if we wanted to control the rate? How might we design different variants of the reaction with different rates? And so this is an experimental paper. We love experimental papers because they show us this, they show us the inputs. They are experimental, so they tell us what the outputs are. And then what we're hoping is that our tools will be able to predict what the outputs are from the input. So this particular paper had 36 different reactions that it was reporting on. And what they did was that they started with a DNA strand displacement reaction. They tell us what the sequences are. And then they introduced mismatches in different places along the area for branch migration. I've just put a little purple diamond as one example. So they moved that purple diamond along and they get different variants of the same reaction. And they also varied the length of the toehold. So it could be either 4, 6, 7, or 10, say in here. So they have 36 reactions in total, and they reported on the rates for these reactions as a function of where the mismatch is. And roughly speaking, the further away the mismatch is from the initial toehold, the faster the reaction is, because the mismatch is kind of less disruptive. When the toehold initially binds on, if the mismatch is right there, the invader is more likely to fall off. But if the mismatch is further down the branch migration, then things proceed smoothly and the mismatch doesn't really have much effect. So that's a way you could control the rate of the reaction, by placing this in a strategic way. And what they claim in their paper is that reaction rate constants can be tuned across 3 orders of magnitude by introducing mismatches without significantly changing the stabilities of the reactants or products. So that's a nice feature for a DNA strand displacement reaction designer. The second example that I'll describe is a very simple reaction of DNA hybridization. And again, it's data from a paper that was interested in the rates of DNA hybridization. And they studied 47 reactions where they took a 23-mer, so a strand with 23 nucleotides, and then the complement. And they just put the 2 strands in the test tube, and they should hybridize and form a duplex. And they carefully designed their strands so that they shouldn't really fold into hairpins or do weird things that would be very stable and slow down the reaction. Because other papers had studied the impact of stable hairpins in slowing down or speeding up association or dissociation of helices, and they were interested in what happens if there aren't such features in the strands. And again, of course, in addition, I should have said the first time, in addition to the sequences, the initial structures and the final structures, you have to specify the concentrations, the temperature, the salt concentration, and so on. So all of that goes into the prediction method. So in this case, again, the paper reports 47 reaction rates, and the paper hypothesizes as to why the reaction rates are the way they are. So you'd like to have a way to predict, or you'd like to have some kind of mechanistic insight as to what's going on. And in this case, the rates did vary by quite a lot, and that was a bit of a surprise. And so they claimed that even though positive delta G secondary structures aren't very stable because the free energy is positive, they say this paper hypothesizes that they can still change the hybridization rate by 2 orders of magnitude, even for 23 mers. So that's quite a big range of rates for very small, simple molecules. Okay, so the problem is you're given the inputs, you want to produce the rates, or ideally some insight. So how do you do that? What computational method are you going to use? So a very standard method is to sample folding trajectories. So you, for example, start with the initial configurations or structures of the reaction, and then you generate a folding trajectory. So you keep adding and removing base pairs, and you see how much time that would take, and you see where you end up. And there's different variants of this. I've listed a few papers here. Some papers include pseudoknots, some don't. Some actually add some molecular dynamics information, and some will only focus on folding a single strand, and some will handle multiple strands. So there's different variants of the same general idea. And I'm going to focus on multistrand, one of the references on the last slide, because multistrand handles multiple strands. That's why it's called multistrand. And I'm interested in three-way strand displacement where you got 3 strands. But the principles are really very similar to one strand. So what you have is a continuous time Markov chain model. And the authors of the original multistrand method are Joe Schaefer, Chris Thatchuk, and Eric Winfrey. And some of you might remember Chris too. I'm sure he gave a student talk here a long time ago. He was also a student in the Beta Lab where we worked, and he got his Ph.D. in computer science, and then he moved down to Caltech, and now he's in a computer science department, but he's got a wet lab, so he's doing biomolecular computation in a computer science department. Okay, so here I'm just going to, hopefully, if this works, yay. This is what like simulating a folding pathway looks like. And this is from a different piece of software because there's only one strand called Kinefold. Not sure if I can run it again. Try that. But you can see base pairs are being added and removed, added and removed. The strand initially has no base pairs, and gradually more and more base pairs get added and removed until it finds its way to a stable configuration. So that is what a folding pathway is. And you're interested in the time for each of those elementary base pair addition and removal steps. And you would add up all the time. Now, of course, if I were to press this a third time, you'd see exactly the same folding pathway. But really, every time a molecule folds, it's stochastically following its own random pathway. So it's not going to be the same pathway every time. And that's why we have an underlying Markov chain model. And in this model, the states are, we represent them as secondary structures. And 2 states are connected if you can get from one to the other by either adding or removing a base pair. And there's going to be a certain rate at which that base pair gets added or removed. These rates are calculated from underlying thermodynamic and kinetic parameters. And this overall Markov chain will satisfy detailed balance. It's just a basic biophysical condition so that in the end, if you were to simulate this long enough, you would be very likely to be in the equilibrium or minimum free energy state. And the way I've depicted this here is that, of course, each of these secondary structures has a free energy, so you can imagine that they're situated on a free energy landscape that has basins and saddle points and high peaks. And a sample, if you're going to sample a pathway from the initial to the final states, it's trying to find the lazy way that avoids high energy configurations, guided by randomness. So it's biased each time it makes a decision about what's the next state to visit. It's going to choose, it's going to favor the ones that are lower in energy. That's just how it works. Okay. So then all we have to do, given our inputs to produce our outputs, is like do so, say, 10,000 stochastic simulations. Each one will have an associated time. We can plot those times to get our time distribution, and it's easy to calculate the rates from the time distribution. So the big challenge here, there's lots of challenges. The one big one is that, unfortunately, this landscape has a number of states that's exponential in the length of the molecules that you're feeding into it. So you can't really just build this Markov chain and do the simulation. Of course, the simulation can also be very slow because if you get trapped in a basin, you're just going to be spinning your wheels and spending a lot of time in the basin, and it could be very slow. A third problem is that the rate predictions are often inaccurate, and that's because although there's been a lot of work on calibrating thermodynamic parameters for DNA and RNA prediction, there's not been so much work on calibrating kinetic parameters. So if you've ever used the Vienna package or RNA fold or any of those other programs to fold an RNA molecule, they're not too bad, they're pretty good. But because the thermodynamic parameters have been refined for decades, but the kinetic parameters is another ballgame. We don't know how to do them. And multi-strand, only the initial inauguration of multi-strand has thousands of thermodynamic parameters. It has 2 kinetic parameters. So that's a pretty crude model of kinetics. And then finally, now you've got your 10,000 extremely long folding pathways that you generate using the software. Now you've got to make sense of it. You want to say, oh, wait a minute, they're not going to fold the way you thought, this hairpin forms. You've kind of got to extract that information out, and there's no support for that provided. So this is the multi-strand software that we are collaborating on and trying to improve the methods. So this is just a slide to show you where we're at. I've talked about the 2 examples, 3-way strand displacement and hybridization. And then I've talked about the methods, which is simulating folding pathways through an energy landscape modeled as a Markov chain. And now I'm going to describe a few things that we've been doing to hopefully improve this process. So first of all, we've proposed a new Arrhenius kinetic model that improves the 2-parameter model that was initially in multi-strand. And in our model, the transition rates from one secondary structure to another can depend on the local context around the area where the base pair is being formed or broken. And so I've highlighted those contexts in red and green here. There's a left context and a right context. So for example, if a base pair were to form between 2 stacked pairs, that might have a very fast rate, whereas if that base pair is forming, like in this situation where there's an open loop off to the right in green, maybe it's a bit slower. And the original multi-strand wouldn't distinguish between those 2 cases, but the new Arrhenius model will. And specifically then, the forward and backwards rate for an elementary step can depend on an activation energy and also on a scaling factor that is dependent on the left and right contexts. And the current model independently treats those 2 contexts, half contexts, independently. So we in our model, we consider 7 different half contexts, and each has an associated activation energy, and there's a scaling factor that's needed. So we have 14 parameters for unimolecular reactions plus a bimolecular reaction. So we're up to 15 parameters from the original 2 in the original multi-strand. All right, so we know how we want to model things, but we don't have any values for those parameters, right? So we have to figure them out. So what we're going to do is Bayesian parameter inference, which is kind of the obvious thing to do, I guess. We started with maximum likelihood estimation and then have moved on to do more rigorous Bayesian parameter inference. So we have our 15 parameters. We start with a very vague prior for those parameters. We have a training set that we had to curate ourselves. It's got over 1,000 DNA reactions of different types. And then we're going to learn better parameters. We're going to get a posterior distribution on our parameters, which are theta in this slide. And we're using Bayes' rule to do this. So we'll start with the prior, and we'll see what's the likelihood of our training data given the prior. That will give us a posterior, which should be better than the prior. And then we'll repeat this over and over and hopefully get to a good set of parameters. So conceptually, this is fairly straightforward. It's kind of the natural thing to do. So the challenge is likelihood estimation. So for that, we have a particular, we've chosen our kinetic parameters theta. We've drawn them from our prior distribution. And now we have to basically run our multi-strand simulator for all of our 1,000 reactions, which I said was extremely slow and painful to do. And so that would take forever. We'd never be able to make any progress on our Bayesian estimation. So we have to come up with some sort of approximation for our Markov chain. And so initially what we've been doing is truncating our chain. So we're just removing a whole lot of areas where we think the pathways are very unlikely to visit and just focusing in on a much smaller area, which we find using somewhat bias sampling and then elaborating around those bias samples. And we get a much, much smaller Markov chain, and then we can use exact solvers on that Markov chain. So instead of doing our Gillespie simulations, we can actually use matrix equations and get the answers. And then the problem we ran into is that these exact solvers often don't work because there are numerical issues, because these matrices are multi-scale. Some of the transitions are extremely fast, some of the transitions are extremely slow, and these matrix methods just don't work very well on the kind of matrices that we have. So we've made some progress this way, and we have gotten better parameters, but it's been a challenging process. So the last thing I'll mention, so today, I mean, we're still working on this inference. We need better coarse graining methods for Markov chains, and we need to improve all of that. We need better Arrhenius models eventually. But for now, we did improve our models, and so we'd like to use our new and improved multi-strand with the better Arrhenius model to get insight on some of the reaction systems of interest to the community. And today I'm going to talk about a little study we did on the DNA hybridization kinetics. So we'd like to know, like, what might account for the fact that there's 2 orders of magnitude rate differences to be found among these 47 reactions. So I'm going to show you a plot here. And in this plot, the 47 reactions are numbered along the x-axis, and they're ordered by decreasing rate. Okay? And what my student Jordan did was she partitioned up the whole state space into groups. So some of the states or secondary structures in green are the ones that you might find early on in your folding pathway simulation, where there's very little structure to be found at all. And the second little rectangle in the bottom is a pink one with an M. So a mishybridized stack forms. So maybe the 2 strands, which are initially floating around in solution, maybe they find each other and bind, but they don't bind in the right way, they bind in the wrong way. And so you get a misstack. Or maybe a misstack forms, and in addition, a little hairpin forms. So all these different things can happen, and she partitioned up the state space into just 8 possibilities, which is a sort of a human interpretable way to think about what might be going on. Is a hairpin forming? Is a mismatch forming? Or, you know, does the reaction precede a property? And then she did many simulations, and she plotted in a normalized way, like what fraction of the time is spent in each type of state, assuming that the trajectories end in a fully hybridized, the product is what you want, fully hybridized, for these 47 reactions. And so you get this really nice little picture where you see a lot of green on the bottom, because initially there isn't much structure. The 2 strands haven't bonded with each other yet. And then you start to see a lot of pink maybe. So these mismatches really do form. And then ultimately you get to the gray, which means that a proper stack has formed. And once a proper stack is formed, the rest of the strand sort of zips up and hybridizes the way you want, and the reaction ends. But you can also see that there's quite a bit of variation here. There's one odd reaction, number 46, where it's all yellow. So you have a hairpin and a stack, and it's just an intransigent hairpin that won't break, and so that's really affecting the reaction. And there's one with a deep purple where you have the mismatch, but you also have a hairpin. So there's a bit of variation there. And then she also plotted the same thing among trajectories that end in dissociation. So the two strands come together, they might hybridize, but they might come together and then break. And so in your simulations, that's often the outcome. And she did the same thing there. And as you can see, when things end in dissociation, usually like a good gray proper stack never really gets a foothold. Instead, you've got a lot of mismatches, but you can see again in reaction 46, for some reason, the hairpin seems to be very dominant along with a proper stack, but it screws everything up for the reaction. And one of the main conclusions we had from looking at this, you can also see, say, number 39 has a lot of purple. So there's no real nice trend. You can't really say there's an easy way to predict what's going to happen. Like each strand is its own character. It has its own quirky way of folding. And it's really hard to come up with a nice theory about what goes on. There's blue appearing on occasion a lot of blue appearing in some cases, like a hairpin, and not in other cases. So each strand, it's just difficult to see a nice, easy explanation across the whole set of molecules. Okay, so with that, I'm going to move on to future work. And as I mentioned before, in order to really be able to handle more complicated reactions like three-way strand displacement, which we're kind of able to do now a little bit, but eventually we want to get to four-way branch migration, we're going to need to beef up all of the things I told you about. We're going to need better ways to coarse grain our extremely large, exponentially large Markov chains, and we'd like to improve the Arrhenius kinetic model, which now has 15 parameters, but there are lots of interesting ways we could improve it. Of course, the challenge there is we don't have a lot of data, so we have to be very careful to make sure that the model is consistent in scale with the data that we have to do the training. Our focus has been on DNA because that's what people in the DNA computing community care about. But all the principles would apply also to kinetic prediction of RNA. And RNA, as we saw in the example of Peng Yin's RNA switches, is quite interesting to synthetic biologists. And here's a paper that actually is taking a completely different approach to our approach. It's taking a deep learning approach as opposed to a biophysical modeling approach. And it's an approach also to predict, as you can see in the part that I've highlighted, they want to predict the behavior of their synthetic biology components. So they want to be able to predict kinetics of engineered RNA elements. So think of it like a little RNA switch or something. You want to predict how it's going to work. And they say that remains a challenge. They've invented this deep learning approach. They have incredible resources, so they had a huge data set, which is awesome to do their training on. And they say that their method outperforms state-of-the-art thermodynamic and kinetic models, so the kinds of models that we're interested in. So, but I'm mentioning it here because actually, I don't think they really did a great job comparing with the state-of-the-art thermodynamic and kinetic models. I think there's still a lot of potential for these models, especially on small RNA switches, to be able to give really good predictions. And so I personally like biophysical understanding, so I want to keep pursuing this. But I think that eventually there will be synergy between more deep learning approaches and biophysical models. I think they will converge eventually. But the larger point is would be super interesting to do this for RNA as well as DNA. And then finally, what about RNA-DNA hybrids? So this paper that I'm referencing, which just appeared recently in PNAS, points out that in many biological processes, what you've got is an RNA molecule invading a DNA double helix to form a hybrid. And wouldn't it be great if we could model the kinetics of that, and if we could control the kinetics of that? And it's super interesting that because DNA base pair free energies are different in some sense from just RNA and DNA. What they're pointing out is there's huge potential to fine-tune the kinetics of these reactions by modifying the sequence, permuting bases in the sequence. And again, you'd really like to have a software tool that would predict the kinetics of RNA-DNA hybrids if you were going to fine-tune the rates at which these hybrid reactions happen. And then, again, long ways down the road, all the reactions I've talked about are really quite small strands, short strands. But DNA origami is a really cool biomolecular computing technology invented by Paul Rotherman back in 2006. So he took an extremely long strand, a viral strand, over 1,000 nucleotides long, and he programmed up short staple strands that would bind to two different parts of the long strand and pull them together. And he was able to build smiley faces out of DNA using this idea. And this was back in 2006, and the field of DNA origami has exploded. You can find all kinds of amazing maps of the world or fractal images or all kinds of cool stuff. But now we're talking about structures that are highly pseudoknotted and have thousands of nucleotides. And you can find even videos. Nobody really knows, like, what's the order in which the staples bind to the long strand and really what's going on. But being able to, like, simulate that and predict what's going on would be really cool. It's kind of amazing that it works so well, given how little we understand about the details of the folding process. Okay. So in closing, I hope I've conveyed a little bit of my enthusiasm for the field of biomolecular programming. I guess my view is that computer science concepts are beautiful and they transcend current technologies like silicon-based technologies. And in the future, we'll see these concepts of computer science being relevant and important well beyond current technologies, and hopefully in cells where we can come up with neural networks and put them in the cell to do something hopefully good, and other wet environments like mediating issues in a wet environment. I personally have enjoyed the field because it's a rich source of computational modeling and analysis problems. And then these can be relevant to other domains too, which is nice. It's very creative. It's an interdisciplinary field. Because there's no killer apps yet, it's a small field and very collaborative. Like, I work with chemists, I work with biophysicists, and we kind of rely on each other. There's a real spirit of collaboration that I didn't ever find in computer science, but I really love this field because I find it to be a good place to work. So I hope you might also enjoy it at some point. And so thank you for listening. And I just again want to thank the many students both at Caltech and at UBC who have contributed to the DNA kinetics prediction work. Thank you. Happy to take questions, or we can move on to beer.
I'll just serve beer.
Oh, okay. Oh.
Hi. So I've heard about biomolecular computing in the sense of programming signaling molecules and reporter genes. do you think that there's any potential to combine DNA strand displacement with these reporter genes, perhaps by, I don't know, manipulating the DNA strand to be a repressor sequence or something like that?
I do think so. So the question is, could we combine the principles of DNA strand displacement with goals of producing reporter genes? Okay. And so I'm not sure exactly how the reporter genes are being produced, but I think Peng Yin's example that I gave, like let's suppose you want to produce a reporter gene when certain environmental conditions hold. And maybe those conditions are kind of a complicated logic expression. Like either this pathogen or that pathogen is present and this other thing is not present or something like that. So that's where you would introduce it might not be DNA, it might be RNA, or it might be some other, synthetic variant. But yeah, the principles, I think, should be relevant. Does that make sense?
Yes, that's really exciting. Thank you so much for your talk.
It's my turn. Thanks for the talk, Anne. So I might have missed this, but I'm just wondering in the wet lab, what would be the, when you're trying to, what would be the readout essentially? And you mentioned that data, acquisition is a problem and having enough data to parameterize, or I would imagine if you want to do deep learning, it also requires lots of data. So just wondering, how is that problem going to be solved, or what's the improvement over the years?
Yeah, so the data we typically work with, look at a three-way strand displacement reaction. So what happens is then this target strand is produced. And if you put a fluorescent quencher on that strand initially, when it's bound to the incumbent, then you don't get a signal. But as soon as it falls off, you start to get signal. So that would be a typical way that we would get
Okay.
we would then measure that over time, and that would give us the rate.
And that would just be a single readout? In other words, there's no sort of
Yeah, it would
dynamics
just be,
in the
yes,
sort of,
correct. It would just be a signal readout over time as the reaction progresses, but we would get no insight as to what was actually happening. So we're hoping that then that
because
our
I'm
model
just
is getting
thinking
that
of,
right.
MinION, for example, or some sort of molecule channels. Is that something possible? Just thinking
Yeah,
outside the box.
I do think so. I think that, if you pull molecules through a nanopore or something,
Nanopore,
you should be able to know more about what's happening in that process. Of course, that's a very different process where the physical force is being applied compared to, the molecules interacting in a closed environment.
Yeah.
But I think that kind of data should be useful.
Yeah.
We don't use that kind of data at the moment. All we get is the rate at the end. Yeah, but sometimes you can look at the curves, like if you look at what's happening over time, and the reaction is proceeding, and then something happens and then it slows down. There was some structural mechanism in some sense there's two modes to the curve, and to look for two folding pathways. So sometimes the fluorescent curves give you a clue as to something interesting must be going on. So I guess the point there is often these papers only report one number, but if they give you the whole curve, that's way more valuable.
I'll try to ask my question, but I think I'm not going to use the microphone. So this might be a naive question in this realm, but when you interfere with the shape of the DNA, you kind of do interfere with the function of it as well. So when you introduce those kind of developments or those kind of attachments to the DNA or hybridization to the DNA, do you also interfere with the viability of the cell? And does that, let's say we are trying to make a computer here, right? We want to use it more than just one time. So how would that impact the cell's life cycle, I guess, my question is.
Yeah, I think that's a bit beyond my pay grade right now, honestly. We're just trying to get simple logic circuits or neural networks working in an extremely controlled environment for the most part. And then there are some really amazing and creative and ambitious people like Hong Yin and Niles Pierce at Caltech and others who are taking it that step forward further. And actually, Chris Thatchuk, my student now, he's working, putting things into blood samples. So that's not really putting it into the cell, but it's more of a safe environment. And there's a lot of challenges, like how do you get the molecules through the membrane into the cell? And then how do you make sure they're not instantly degraded by the cell's defense mechanisms? And then how do you, as you say, make sure that they don't do anything bad once they get in? And I don't have the answers to any of those questions, but they're all really important questions. Longer term.
Thank you for your great work and all the information. My question is, what criteria were you used to determine that mediation was related to the biological matters rather than the model limitations?
So the question is, what criteria do we use to...
What criteria did you use to actually analyze and figure it out the mediation is related to biological matters or model problems, limitations?
Okay, so I think, to be honest, again, our work is not situated in a biological context like a cell. So we're assuming that the molecules we're trying to understand are in a much more controlled environment. And that's hard enough. And I think it would be much harder to figure out if you were to put them into a biological environment than what's happening. But I suppose if I look at people who are putting, say, a simple DNA logic circuit into a blood sample, at some level, that is a biological environment. And so, but it's a simple-ish biological environment. It's not a living environment, but it's got signals, does somebody have a particular disease? You're looking for whether a signal is there or not. So it's that kind of work, again, is ambitious and hard, but I think you would have to be sure that the molecules you're introducing are unique and, they will, say, be fluorescently labeled, or they will have some particular way of signaling. And, you'd have to do control studies. You'd have to do things like that to make sure that they're working the way you're hoping. I do encourage you to invite Chris up someday to give a talk, and he should be able to answer some of these questions better.
Okay, I think I'm next. For the reactions, I think the strand displacement reactions, was one of the parameters that you've investigated the concentration of the base strand? I'm thinking in terms of like length and just like number of different strands when it's already bonded. So when there's when there's the incumbent is still there on the base strand, so it's double stranded. How many of those in the test tube at once? And I was wondering if you could talk a bit about that.
Yeah, so the multi-strand model is very simplistic, and it assumes that each of the three molecules starts off with the same concentration. So there's equal numbers of the three different strands. And then the reaction starts with a bimolecular step where two of these molecules will bind to each other. And so they use that concentration to determine the rates of the bimolecular step. And then once the two strands are bound, it's kind of unimolecular, so the rate doesn't really matter anymore. But it's still not ideal. Like you really would like to have, what the multistrand is not doing is tracking changes in concentrations over time. Right? So in many situations, you really would want to do that. And that would be another enhancement to the method that is currently.
Question. Oh, sorry. Thanks for the great talk. You showed us the folding simulation animation, and I think your simulators assume that the folding process is stochastic. Do you think it is also stochastic in the biological world, for example, in the cells, or is it just assumed to be stochastic for the simulation? And more importantly, do you think if it's stochastic, do you think it always converges to one folding structure?
Okay, so I absolutely think it's stochastic in the cell, too. A big difference between, say, RNA folding in the cell and the way I talked about it is that RNA folds co-transcriptionally in the cell as it's being synthesized. And as a result, parts of the molecule are going to fold possibly into quite a stable structure before other parts are even synthesized. And that could mean that the ultimate structure is not the minimum free energy structure, but is some other sort of stable structure that's consistent with co-transcriptional folding. I think our bodies are amazing and they work pretty well. So I think almost all of the time things are folding the way they should. And I think the cell has all kinds of amazing mechanisms to deal with the bad actors that don't do the right thing. Great.
Yeah, thank you so much. Let's thank Dr. Condon for her amazing talk. There's going to be pizza outside, so feel free to grab some slices and please enjoy the social events. Yeah, thank you. Thank you so much. It was wonderful.
Okay, good.
Yeah.
It was fun.
Thank you.
There's no beer anymore. The last time I gave a talk was on. That's okay. I don't need a beer.
I'm sorry about that. Yeah, we are not allowed to serve anything alcoholic anymore.
Everything gets way more.
Yeah.
Okay, welcome. Thanks for the good question. That's a good question.
First of all, the splicing and the folding effect splicing.